Welcome, good afternoon, and thank you for joining us for this Flinders University Meet the Minds for what I think is going to be a very fascinating topic on how we use wastewater, sewage, in the fight against the COVID-19 pandemic. My name's Alan Sicolo. I'm the Senior Marketing Manager here at Flinders University, and I'm really excited to host today's event as we explore the public health response to the COVID-19 pandemic, where we've been able to test sewage in our waterways to uncover the SARS-CoV-2 virus, where it's been excreted in bodily fluids of both symptomatic and asymptomatic cases. This seminar will discuss the application of the technique and its contribution to the management of the pandemic. I guess to begin with, before I go on, I would like to acknowledge that we are hosting this forum on the traditional lands of the Kaurna people, and that we pay our respects to their elders past, present and emerging. We acknowledge and convey our deep appreciation to the elders of all the nations upon Flinders, in which Flinders University operates. So this event is part of our Meet the Minds lunchtime lecture series, where we give you the opportunity to meet some of Flinders University's most engaging minds as they bring you the latest research from a diverse range of fields. And today I'm quite excited because we're fortunate to hear from Professor Howard Fallowfield. Professor Fallowfield is an aquatic microbial ecologist. His research focus is health aspects of water quality, which have been conducted across a range of aquatic environments, including drinking water, wastewater and recreational waters. Howard has over 25 years of research experience in the UK and Australia, both designing, constructing, operating and evaluating the performance of pilot plants for both drinking water and wastewater. As always, we are keen to make this an interactive event with live Q&A sessions. That's your chance to participate in the discussion and pose questions to the speaker and to Howard in real time. We do ask, however, that everyone please treat this forum as a place of respectful engagement, where people are treated with dignity and where differing views are tolerated. So I think we're ready to start receiving questions now via the messaging function on the Teams platform, so please feel free to ask questions. And so now it is my pleasure to invite Professor Howard Fallowfield. Thank you, Professor. Over to you.
Thank you, Alan. And I'd like to thank the Meet the Minds team for inviting me to participate in this lunchtime lecture series. So we're going to talk about the role of wastewater surveillance in managing the pandemic that we're all currently experiencing. And I'll give you a bit of an overview of the virus, the SARS-CoV-2 virus. It's a single-stranded RNA virus, and it has the spike protein that we've all heard about that causes the attachment and infection. It's got an envelope. It's an interesting virus. It's got an envelope protein as well, a capsid protein as well. It can exist in water. What's important to understand the concept of wastewater-based epidemiology is that the picture A is an intact virus, a virus that if it comes into contact with a susceptible individual would cause the COVID-19 infection. But it can also be incapacitated in water where it starts literally to fall apart, and then it can be completely degraded. The important point is, though, that the single-stranded RNA can be still available for detection in all of these three conditions. And that in part explains why you may have heard on the media when reports of detection in wastewater have been described as fragments of the virus have been detected, because we detect the RNA material, the nuclear material. We don't know whether that material is coming from an intact virus or it's coming from an incapacitated or degraded virus. So the organism is a single-stranded RNA. It's relatively unstable in wastewater. It fortunately quite rapidly degrades through wastewater treatment plants, so it's not a big risk of potential infection from wastewater, which is good news for workers in the wastewater industry. Way we detect it, we're all now molecular microbiologists with PCR tests for the nasal swab, etc. And we use the same technology in terms of the RNA being reverse transcribed into cDNA, and then we use a standard polymerase chain reaction to amplify that DNA. Briefly, what this technology enables us to do is that we may have very few viruses present in the wastewater. It'd be impossible to detect using any other method, and via this PCR technology, we can rapidly multiply up the gene that is genes that is associated with the SARS-CoV-2 virus. So these genes are very specific to that virus. We can, using PCR techniques, we can rapidly multiply them to a point where we may end up with billions of copies, starting from a very low number to a very high number, which then enables us to detect the virus at low concentrations in the wastewater. We're fortunate in that the virus was made available very quickly. It can be killed via gamma radiation, still enabling the RNA to be detected. So we can prepare a standard curve of viruses of known concentration, and from that standard curve we can determine the numbers present in our wastewater sample. The important thing is we don't determine it as numbers of virus. We talk in terms of copy number, because of that uncertainty about whether the virus is intact, incapacitated or degraded. So in this talk I'll be talking about copy numbers rather than viral numbers. So we're going to be talking about wastewater. So wastewater is sewage, as Alan referred to it. It's a combination of water that comes from bathrooms, laundries, showers and toilets. So there's a large variety of bodily fluids that go down into the sewer, and work so far looking at where the sources may be are saliva. So when you clean your teeth and you spit into the wash hand basin, evidence is that there's fairly large numbers or copy numbers of SARS-CoV-2 virus produced in saliva. Again, microbiologists, microorganisms are usually exist in very large numbers in the environment. And even when we talk about copy numbers, copies of the SARS-CoV-2 gene, the numbers are very high. So I'll be talking in terms of 6 to 10 log cycles. So that means 6 means 1 million organisms per mil. 10 means 10 billion copies of the gene per mil. So we're talking in very large numbers. So you can see here, 2 days after onset, there's a very large numbers of copies coming from saliva in a study from South Korea. Similarly, a large number of copies from Hong Kong. So we know that it's present in saliva. We know that coughing and discharge in sputum, there's very large numbers, billion copies of the virus in sputum. Less apparently coming out in urine from work done in China, Germany, France, in Europe in basis, and South Korea. So the next obvious one that we would expect to look at is feces. So persistence of viral shedding in feces. So this is really trying to determine from clinical samples whether virus is being discharged in feces. And there was a rapid review last year that looked at this from a variety of countries, and they looked at participants that have been clinically tested as positive. That would be nasal swab, PCR, etc., the gold standard. And they found that the numbers of participants range from 1 to 206 in these studies, and the age range was 3 months to 87 years. When we talk about these bodily functions, we have to accept we're talking feces, we're talking anal or rectal swabs in patients to determine whether there is virus present in the fecal material, and over 50% of the samples were tested positive for fecal SARS-CoV-2. The question then is, as the disease progresses, how long do you continue excreting virus in the feces? And again, 1 week after symptom onset, there was over 10 million copies per gram of feces detected, decreasing over a period of time to only 1,000 copies, but still significant. So we know it's still present, and the question is then how long does it remain present? And the duration of shedding in this review was between 1 and 33 days after testing negative. So this is when the patient would have been declared free of the infection as from the nasal swab, but it was still present in their feces after between 1 and 33 days. The issue with our detection of genes through copy number is that we actually don't know whether the virus, as we've said, is whole and intact. And it's only when it's whole and intact is it capable of causing an infection. So we know there are fragments of the virus present in the feces in quite large numbers, but we don't have a sense of how infective or how infection may be progressed from contact with contaminated fecal material. And this is where we're looking at whether the organism is alive or capable of infection in a human. And again, after a relatively small sample, we've got 44 positive for the virus. And then we have to use really expensive and difficult techniques of cell line culture to understand whether the virus is able to infect cells. And of that, only 2 out of the 4 specimens were cultured for live virus. So it's suggesting that the virus may be present, or fragments of the virus may be present, but the whole virus capable of infection is in quite low numbers in the feces. So what's all this about? Well, we have very good evidence that these sources of saliva, sputum, urine and feces means that the virus is excreted and could be detected in wastewater. We also have got some idea now about, in this figure, we're looking at the blue is when there was a hospital admission and the duration at when the throat swab was testing positive for the virus, and the brown line is how long the duration of detection of the virus in fecal material. And in this, it's been recognized there's probably 3 groups of shedders, where there's some that have really prolonged shedding in group 1, and then it reduces through group 2 and group 3, the duration at which you could find the virus present in fecal material. So the take-home message is we know we would expect to be able to find it in wastewater. The question was, would we have techniques capable of detecting viruses in potentially low numbers in very high volumes of wastewater? So a colleague, the foundational study that got the world really interested in this was from a colleague in the University of Amsterdam, Gertjan Medema, and they were really ahead of the pack in terms of their thinking about this. And they took a view that they could go to Schiphol Airport in Amsterdam, which was an international airport, and they could start looking at seeing if they could detect SARS-CoV-2 in the wastewater from that airport. And they set about their research well before any clinical cases were appearing in the Netherlands. And they were using this N2 gene, which is characteristic for SARS-CoV-2 virus, and they started to look at it in the wastewater emerging from the airport. And this is a really nice, this is from February through to May. And what we're seeing here is in the brown, it's the detection of this gene characteristic for SARS-CoV-2 in the wastewater. The blue line is reported cases of COVID-19, clinically reported, clinically identified, and then hospitalizations. And you can see there is a really nice agreement between the increasing detection of this characteristic gene in the wastewater, along with the increase in reported cases and hospitalizations. And then as that first wave trailed off, you can see that the gene count decreases, as does the hospitalizations and the new reported cases. So this study was really the one that caused microbiologists and public health experts to sit up and take notice about how wastewater-based epidemiology might be used during the pandemic. Now, wastewater-based epidemiology is not necessarily new, and some of you may already be aware that it's been used to evaluate illicit drugs in populations and in prison populations and in communities. And indeed, in Australia, there's a survey conducted pretty near every 2 years to identify the extent of illicit drugs in wastewater, and by that make some comment on the use in various populations in metropolitan cities and rural communities in Australia. So the idea is that we've got a population, it's excreting wastewater produced from that population is a composite sample, if you like, of both pathogens, disease-causing organisms, and potentially harmful chemicals in that water. It all arrives at a wastewater treatment plant. We could take a sample of that water under various situations and various methods, which I'll talk about towards the end of the talk, and that wastewater sample can then be delivered to a laboratory. It could be analysed for bacteria, viruses, or protozoa that might be potentially pathogenic and cause disease in the community. And we can then make there's a caution here in the public health domain about how good our measurements are and how we should really use this data to extrapolate back into the population the number of cases of a particular disease. And there's some caution about using the data from wastewater sampling to extrapolate it to numbers of cases in the population. But it's a very good method for sentinel surveillance of the prevalence of disease-causing organisms in a population using wastewater samples. So our involvement in this as a research group in Flinders was that the university had some insight into offering research money for an urgent response to the COVID pandemic, and we were fortunate enough as a group to be awarded funding. And that was very timely because in Australia there was just forming this Colossus project, which is the collaboration on sewage surveillance for SARS-CoV-2, and we were able to become a foundational member of this, and it's now extended to be a national research project. And you can see the plethora here of corporate logos, and it really does show that it's across Australia, this interest in SARS monitoring in wastewater. And our role was largely to monitor the wastewater coming out from rural communities, since the big utilities were doing the major treatment plants from major conurbations like Adelaide, Perth, Sydney, Melbourne, etc. And you can see that that you will have seen this in the press, where it's really taken off in terms of taking a water sample, processing it in the lab, cleaning the sample up, using this PCR methodology to detect the virus, and then having some inkling about the presence of the virus in a community. Part of the issue has been, in the early days, the testing was done at the inlets to the major wastewater treatment plants. So in South Australia, that would be Bolivar, Christies Beach, Glenelg. Bolivar alone takes wastewater from a population of 700,000. So it was very quickly recognised that that was not sensitive enough to say anything about the smaller populations. So increasingly now we've moved up the catchment and we've gone to smaller population sizes and taking samples from smaller population sizes. So for instance, in Adelaide, we're now, SA Water are now sampling some 22 sites, I think, and the population size varies from 12,000 to 50,000. So we get a much more discrete analysis of populations and an early warning of presence of SARS-CoV-2 in that population is now possible. As Alan said, the advantage of wastewater-based epidemiology is it detects both asymptomatic and symptomatic cases. It can be used to evaluate the progress of an outbreak. In Adelaide, during the festival period, when we had increased detection in one particular catchment, it was publicised, and it led to almost a doubling in population going out to be clinically tested using the nasal swab. So it led to a public health response in that context. In the future, it might well be used for mass gatherings, for instance, stadia where we've got sporting events, aged care, halls of residence in university environments, for instance. It's certainly being used on marginalised communities, the APY lands, indigenous communities, etc. Some of the issues of wastewater-based epidemiology is resourcing, the cost. The idea that you've got to do traffic management to access some of the sewers that you want to sample can be very expensive. And the technique alone in laboratory terms is about $400 a sample, but that's still extremely cost effective compared to clinical testing. There's been some concern that you've got to be careful that you don't stigmatise already disadvantaged communities. There has arisen legal issues, who owns the wastewater when you're sampling private locations, and the Department of Health had to work through the response, and they rapidly realized they had to be transparent. You can go on websites now and you can look at the testing outcomes for various communities in the various states. So this is a truly national response, and there are some concerns about well, not concerns, but there's some ongoing research needs. We obviously don't know everything about wastewater-based epidemiology in this context, and part of it is about the shedding still needs to be understood, the sensitivity of the assay in terms of return of false positives and false negatives, how we do the modelling maybe to get back to potential numbers, and how many people in a catchment have to be positive for us to be able to get a positive detection in the wastewater, and standardising methods, etc. One of the areas that we're really working on at Flinders is that one of the questions is, how do you get a representative sample of wastewater where you've got if you think about how you use the toilet and when you go to urinate and defecate, that influences when the virus is likely to be present in water. So currently, initially we started with grab samples between 8 and 9 o'clock, because that's when everybody gets up out of bed and goes to the toilet, and then it takes a bit of time for the wastewater to appear at the treatment plant, and that was taking samples simply with a bottle on the end of a pole. We then rapidly moved to the idea that we could have an automatic sampler, refrigerated. You can take samples out of the sewer at any time. You can combine samples taken at different times and get a composite sample for the day. But increasingly, a colleague at Monash University, and you may have seen the news items about the sewer submarine, Dave McCarthy, developed a simple sampler that's down in the bottom right here, where you put some absorptive material in a plastic container, deploy it for 24 hours and get a sample that's integrated over that 24-hour period and maybe much more representative about the presence of the virus in that population. And Flinders have now got a research project with our nanotechnology group, where we're looking at developing smart materials that may selectively, hopefully, absorb SARS-CoV-2 or any other viruses or bacteria, to adapt the technology to develop wastewater-based epidemiology for monitoring other potentially pathogenic organisms present in communities and using wastewater for that detection. So we think it's a really exciting area. It's great that the Departments of Health have realised that this is a valuable tool to look at infections in communities, and we think it will develop further in the future. I'd like to thank my colleagues who have been involved in the projects we've been involved in terms of monitoring rural communities. We're all happy to report we have no results, which means that the samples that we analysed for these rural communities all returned negative results. So once again, I'd like to thank you for your time, and I'm happy to accept questions. Thank you, Alan.
Thank you, Howard. Wonderful and really insightful, and I certainly do encourage anyone who's got any questions to use the chat functionality. But certainly to get things started, Howard, your research and the techniques developed are obviously critical and have had a direct impact into all our lives, into the community, which is something you must be very proud of. But I'd love to know actually how you ended up here. Why did you choose aquatic microbial to become an ecologist? What drew you to it, and what continues to draw you to it today?
Well, I think we all depend on water, and we interact with water in many different ways, and the microbial ecology works both from the point of view, it can have effect on us both adversely from the point of view of coming into contact with pathogenic microorganisms, and we can also use microorganisms for benefit for wastewater treatment and removal of harmful materials, etc. So it's really an open canvas, and it's got exciting opportunities, not just in microbiology, but also in aquatic chemistry. We have to be across those disciplines of microbiology, chemistry, physics, etc. And it's to me endlessly fascinating.
With that in mind, then, I mean, what's the next big research challenge in the field of water quality? What's the ambition?
Well, I think it's never static. I mean, we now have a whole series of emerging chemical contaminants, and we're all familiar with those that make headlines. I think that's a challenging area both for wastewater, drinking water, and surface water. And in my, I suppose, substantive research in wastewater treatment for rural communities, I think the work we've done in South Australia really has a role to play in meeting sustainable development goals and hopefully contributing to providing safe sanitation for the majority of the world that does not have access to safe, secure sanitation. I'd like to think that's where we can make a contribution in the future in adoption of the technologies that we've developed.
Certainly, a very wonderful and critical contribution. So then, thinking about that a bit even further, then, for anyone that's listening today that's maybe considering a research career in water quality and health, do you have any advice for them?
Oh, I think the advice I would give, as you might detect, is to follow your passion. And I think in terms of engaging with water, as I've indicated, it's microbiology, chemistry, physics, engineering. All those disciplines are really important, and equally it's really important, I think, to keep a broad horizon, in that you really need to interact with other disciplines and you need to understand their language and their lexicon of terms. So I think the broader the better in terms of your education to interact with the water industry, and that includes the social sciences, because we need a social license to operate in this area, and interaction with the population is of critical importance.
We've got a question from Joe, who's asking us if you can talk a little bit about developing smart materials for selective virus absorbance. How close is this, and will it come to fruition?
Oh, how close is it? Only early days. In fairness, wastewater-based epidemiology has been around, and it's had its moments. So it's been used, for instance, to try and get some handle on, we're trying to eradicate polio virus, and it's been used to look at polio virus in communities where this is trying to be concluded. So the early materials are things you can imagine. Early materials in terms of cotton swabs, glass wool. Tampons have been used as absorbent material in these passive samplers, but they tend to be a non-discriminating passive absorption, that the virus gets either stuck onto the material or entrapped in the material. And what we're looking at is that the idea that we could be smarter than that and look at functional groups on materials that would interact with the surface characteristics of the virus or bacteria, if we can move down that domain, and maybe selectively concentrate the virus from within the wastewater and then deliver it to the lab. This has been a huge problem in that the PCR and the detection is quite well developed, and it's quite an engaging area for molecular microbiologists. The area of how do you get the sample out the matrix, how do you get your virus out of the wastewater to deliver to the lab, that is often, to use the word, less sexy research. But it's vital, because we don't really have a good handle on recovery. We need to know how efficient are our methods of getting this virus out of the water. And that's where I think a lot of development can be achieved and improve the sensitivity of the assays, so you could get down to even lower concentrations of virus in the wastewater.
So many more facets to be explored and researched, and I look forward to actually seeing where the research continues and into the future. We are, I think, on time right now, so I want to just say thank you very much, Professor Howard Fallowfield, for sharing your time and knowledge with us today. It's certainly been a wonderful and insightful discussion, and I also would like to thank our audience for the questions and input and interest. And I'll remind everybody that you can watch this session again on the Flinders University YouTube channel or Flinders University Meet the Minds webpage, where you can also register to receive notifications of our future events. I will give everybody a heads up. Our next event is on Wednesday, the 5th of May at 12:30 p.m. On the topic of developing a patient-reported outcome measure for childhood glaucoma, another really important and critical issue. So thank you once again, and please enjoy the rest of your day.